China has identified the cause of the
mysterious new virus.
Coronavirus.
There are fears a rapidly spreading virus has reached Australia.
This is a rapidly emerging situation. There is not a cause for alarm.
The first US case has been detected.
WHO, it has been recorded 219 million cases. From WHO, it has been recorded 219 million cases with 4.55 million deaths around the world. The coronavirus disease, which is COVID-19, is an infectious disease caused by the SARS-CoV-2 virus. The term virus is being associated with a number of plants, animal, and human disease. The mere mention of the term brings to mind serious illness such as polio, rabies, and COVID-19. Viruses are a biological enigma. They have some characteristic of living organisms, such as a DNA or RNA genome, and the ability to evolve and replicate. However, they can replicate by using the metabolic machinery of a host cell only. They do not have a metabolism and they do not respond to stimuli. For this reason, viruses are known as obligate intracellular parasites and are either active or inactive rather than living or non-living.
Classification system for viruses. ICTV has developed a classification system for viruses. It differs from the others because it consists only the taxonomic levels of order, family, genus, and species, and not the higher levels. When the new virus emerges, additional classification is required for a clear identification. For example, influenza A is classified into subtypes based on its glycoprotein spikes, and because of its too, many virus subtypes evolved, causing either seasonal or pandemic flu.
Viruses can be categorized by their size and shape, type of nucleic acid, whether it is RNA or DNA, the presence or absence of an outer envelope. The structure of the viruses can be summarized by splitting the virus particle into two. They are covering and inner core. For the covering, it consists outer envelope and capsid. It is said if the virus outermost structure is viral capsid, the virus is said to be naked. In contrast, envelope virus like influenza virus are surrounded by membranous envelope derived from the host cell's plasma membrane. There are glycoprotein spikes that coded by the viral genome. As the inner core, it consists nucleic acid molecules and various types of proteins. There are a lot of types of the viruses in the world. Here I will show you guys some 3D models of the viruses that had been discovered by the scientists. First, we have adenovirus, T-even bacteriophage, tobacco mosaic virus, and last but not least, an influenza virus. As you can see, there are labels at the viruses that will show you guys the structures of the virus. As you all know, viruses need a host to keep on living. They can't replicate outside the living cell. That is why they need living cells for them to reproduce, because they can't duplicate its genetic material. Virus will hijack the cell protein synthesis machinery to replicate the nucleic acid and the other parts of the virus. This shows the parasitic behavior of the virus. Did you know that viruses attack varieties of cells, but they are host-specific, meaning that some particular virus only capable to reproduce within specific cells? For example, the tobacco mosaic virus infects only plant in tobacco family. For some human viruses are even specific to a particular tissue. For example, the SARS-CoV-2 virus infects cells in lung, and the hepatitis viruses only infect liver cells, and this is what we call as host specificity. So, how does it work? Host specificity is determined by the structure of molecules in the naked capsid or spikes on the virus envelope. The outermost structure of the virus will attach to the receptor of the host cell outer surface and create the lock and key manner. A cell's receptor that doesn't match with the virus can't be infected because the virus is unable to enter.
So let's discover on how does virus reproduce. Not only humans have our own reproduction cycle, but viruses also have their own reproduction cycle, which go down either one of the two routes. Either they want to undergo lytic cycle or they want to undergo lysogenic cycle. Lytic cycle is the most common reproduction cycle among the viruses. Lytic cycle includes 5 stages of reproduction, which are attachment, penetration, biosynthesis, maturation, and release. The lytic cycle starts off with attachment stage. In this stage, the virus will attach itself to the host cell, and then the penetration will occur. The virus will then hijack its genome into the host cell, or the host cell itself will engulf the virus, and then biosynthesis will occur. In this stage, new viral components such as capsid subunits, spikes, enzymes, tRNA, energy, copies of genome will be synthesized. Next is maturation stage. In this stage, new viral components are assembled into new viruses. Lastly, the new viruses will exit the cell by lysis or budding in order to infect other host cells. However, if there aren't too many available hosts in the surrounding, the virus will choose the slower option of reproduction, which is lysogenic cycle.
How does lysogenic cycle differs from lytic cycle?
Following attachment and penetration stage, integration will take place instead of biosynthesis. The viral genetic information will combine with the host genetic information and becomes parts of the host. To put in other words, in lysogenic cycle, instead of killing the host, the phage genome integrates into a bacterial chromosome and becomes part of the host. The integrated phage genome is called prophage. The host doesn't notice the change as the viral genetic info has repressed the gene, which prevents it from being transcribed. The host cell then carries out its normal function and keep replicating without realizing that it is also including the viral genetic information in all of its daughter cells. The prophage is being replicated along the host DNA, and all the subsequent cells, called lysogenic cells, carry a copy of the prophage genome.
The virus stays dormant until once in a while something happens and weakens the virus suppressor gene. The host genetic material then realizes some errors and tries to repair itself by cutting out the viral genetic information. The virus is then released and replicating following the lytic cycle by maturation and release stage.
Here are some differences on lytic cycle and lysogenic cycle. In lytic cycle, the viral DNA does not integrate into host cell DNA. Meanwhile, in lysogenic cycle, the viral DNA integrates into host cell DNA. The virus is virulent in lytic cycle, but in lysogenic cycle, the virus is not virulent. Lytic cycle occurs within short time, but lysogenic cycle takes time to occur. There are symptoms of viral infection in lytic cycle, but there is no symptoms of viral infection in lysogenic cycle.
Animal viruses. Animal viruses reproduce in a similar manner as bacteriophage. However, there are some viruses that introduce their genetic material differently. For naked viruses, it introduced by endocytosis, for envelope viruses by fusion and endocytosis. The viruses later continue to uncoat and remove its envelope once it enters the host cells. They continue the lytic and lysogenic cycle when the DNA or RNA is free.
Highly virulent viruses will directly undergo lytic cycle and causing the destruction of host cell, but different with HIV. HIV will enter the lysogenic cycle first and lie inactive for many years until the symptoms emerge. Other than that, some viruses released by body. In other hand, naked virus released by host cell lysis.
However, retroviruses have slightly different way of reproduction.
Retroviruses are animal viruses with RNA genome that is converted into DNA in host cells by enzyme reverse transcriptase. As example, HIV virus is one of the retroviruses. After virus fused with host plasma membrane, the virus RNA genome is released into the host cell. The enzyme reverse transcriptase in the host cell will convert the virus RNA genome to a single DNA strand called cDNA. The single strand of cDNA is used to make double-strand DNA. Using host enzyme integrase, the double-stranded virus DNA is integrated into host genome. The viral DNA remains in the host genome and replicated when the host DNA is duplicated. This is similar to lysogenic cycle. The difference is it doesn't have repressor genes which typical lysogenic viruses have. So, it is actively transcribed whenever host DNA is transcribed. New viruses will be produced. This is why HIV can remain latent for many years. After that, the new viruses will exit the host cell by spreading.
Some emerging viruses, which is new or previously uncommon illness, are caused by viruses that are now able to infect large numbers of humans. These viruses are known as emerging viruses. Examples of emerging viral disease are coronavirus disease COVID-19. Several types of events can cause a viral disease to suddenly emerge and start causing a widespread human illness, including a virus extending its range or because of genetic mutation. Mutations that occur allow the virus to jump to other species, including humans. Influenza is well known for mutating, and this is why it is necessary to have a flu shot every year. Because antibodies generated from last year's shots are not expected to be effective this year. At least 1,000 different viruses cause disease in plants. About a dozen diseases of crop, including potatoes, coconut, and citrus, have been attributed not to virus but to viroids, which are naked strains of RNA not covered by a capsid. Like virus, however, virus direct the cell to produce more viroids. A number of fatal brain diseases known as transmissible spongiform encephalopathies or TSES have been attributed to prions, a term coined for proteinaceous infectious particles. Prions are proteins that normally exist in an animal but have a different conformation or structure. Like virus, prions, from the normal protein conformation to the prion conformation can be as simple as changing a chemical bond. Once a prion infects tissue, a chain reaction begins that converts normal proteins to prions at an exponential rate.
To conclude, a virus is made up of a core of genetic material, either DNA or RNA, surrounded by a protective coat called a capsid, which is made up of protein. Sometimes the capsid is surrounded by an additional spiky coat called the envelope. Viruses are capable of latching onto the host cells and getting inside them through the concept of host specificity. They reproduce either by lytic cycle or lysogenic cycle. Meanwhile, prions are infectious particles that contain no nucleic acid, and viroids are small plant pathogens that do not encode proteins.